Okay, good morning, everyone. Good afternoon or good night to Fernando. It's my pleasure to introduce Professor Fernando Racimo. Fernando has a bachelor degree in evolutionary biology from Harvard University, a PhD in integrative biology from UC Berkeley, followed by a postdoc period at the New York Genome Center. Currently, he is an associate professor at the section in molecular ecology and evolution in the Globe Institute of the University of Copenhagen in Denmark. In his lab, they use population and quantitative genetics to understand past evolutionary process, with a particular focus on human evolution and ancient DNA. They develop tests to detect patterns of ancient selection, creating methods to integrate functional and population genomic data. And they also infer demographic parameters using present-day and archaic human genomes. So today, Fernando is going to talk about spatiotemporal genomics, and I hope you all are inspired and like it a lot. Thank you very much, Fernando, for being here with us and for accepting our invitation. Help me to welcome him.
Thank you so much, Fabio, for that wonderful introduction. Can everybody hear me okay?
Yes.
Yes, great. Okay. Yeah, it's nice to be back for a research talk. So today I'm going to be talking to you about some of the research going on in my group, where we use ancient genomic data together with some present-day genomic data to trace evolution in space and time. As Fabio said, particularly focusing on human evolution. I just want to talk a little bit about my research group depicted in this picture here. We're located at the Globe Institute at the University of Copenhagen, and we do quite a lot of different things. So we have sort of expertise at the intersection of evolutionary biology, statistics, and computer science, and we like to work a lot with theory, particularly with population genetic theory. There's been quite a lot of developments in population genetic theory in the last few years, especially looking at spatial population genetics and tracing genealogies through space. But we also like to work with data, and there's quite a lot of rich data resources for us to look at, particularly within the field of ancient DNA, where there's now thousands of ancient human genomes, and now more, like also thousands of ancient non-human genomes. I might need to update that text there. So we like to work sort of at the intersection of theory and data within population genetics. And we have lots of different projects. We work a lot on developing methods for reconstructing population histories and detecting natural selection. We like to develop methods for reconstructing population processes in space and time. We work with genomic data, but we also work with ancient proteins, particularly looking for very deep timescales, and we try to apply machine learning methods for doing genomic inference as well. Today, I'm going to be largely talking to you about what we can do in terms of methods for tracing population genetic processes in time and space. And so the driving question for today's talk is going to be how we can model genetic processes over space and time. How can we best understand the evolution of genetic data, accounting for both spatial and temporal dimensions? And so throughout my talk, I'm going to be covering different research projects in my group. And first, I'm going to be talking to you about how we can model ancestry in space and time. We talked a lot about ancestry in the lecture yesterday, and we're going to see how we can use ancient genomes to look at ancestral reconstructions in space and time. We're also going to be looking at how we can better understand natural selection in space and time using ancient genomic data. Also, how we can simulate genetic data in space and time. And at the end, I have a small surprise about a fourth topic that is quite dear to my heart. It's not necessarily related to these first three topics. The exact topic will come at the end. For the start of my talk, I want to talk about ancestry. So go back to the topic that we talked about yesterday. If you remember from yesterday's lecture, you could depict my genome as a mixture of my two parents. If I painted the genome of my father and the genome of my mom in two different colors, and I would have 23 pairs of chromosomes, and each of those pairs would have one chromosome from my mom and one chromosome from my dad. And we remember we had this intellectual exercise where we thought about how we could paint my genome. If, instead of painting my parents' chromosomes, we painted my great-grandparents' chromosomes, which came from different countries in Europe, like Italy and Spain. And these individuals came from different locations. Here's another example of how we can paint genomes to trace ancestry over space and time. This is from a study that looked at Latin American genomes as a mixture of Native Americans, Europeans, and Africans. And so what they did is they collected a lot of population genomic data from lots of different places in the Americas, which are the green circles here. And they used as sort of reference source populations, several Native American groups, as well as European populations and African populations. And what they got when they tried to paint these different genomes for the Latin American individuals was a mixture of Native American, European, and African ancestry. So some individuals had more European ancestry. Each of the columns here is a single individual. And so some individuals have more European or Native American ancestry. Some individuals, for example, in Puerto Rico here had quite a lot of African ancestry. And so you can see there's quite a lot of variation in ancestry throughout the Americas. Now, you can paint Latin American genomes, but you can also paint European genomes as mixtures of ancestral populations themselves. So in the Latin American case, we're talking about a process of mixture of different populations that happened in the last few hundred years after the European colonization of the Americas and the African slave trade into the Americas. But you can also think about modern Europeans today as themselves being mixtures of ancestral populations as well. You can paint the genomes of multiple individuals from different countries in Europe as mixtures of three different source populations depicted here. One of them, it's called early Neolithic, another one's called Western European hunter-gatherer, another one's called Yamnaya. These are very technical terms that are very specific to the field, but essentially what this shows is that in order to model the ancestry of Europeans, we can also use this sort of mixture painting process. And the reason why Europeans are a mixture of different ancestral sources is because if we go a bit further into the past, to the last 10,000 years, Europe as well was formed by a process of, or European genetic variation was also formed by a process of mixture. If we think about Europe about 10,000 years ago, it was mostly populated by different groups of hunter-gatherers. There are some groups that are called the Western hunter-gatherers, another group that is called the Eastern hunter-gatherers. And at some point, and this was a long process that lasted from about 8,000 years ago to about 4,000 years ago, there was a movement of people from the Middle East into Europe that brought farming practices into Europe. These are it's called the Neolithic expansion, the Neolithic population movement. And so for over a period of thousands of years, hunter-gatherer groups mixed with these Neolithic groups over time. On top of that, during the Bronze Age, there was a further population movement of peoples from the steppe, the Caucasian steppe, into Europe after this Neolithic expansion process. And these peoples are thought to have brought herding practices into Europe, possibly also brought the horse herding into the continent. And so Europeans today, in very simplified terms, can be thought of as a mixture of these original hunter-gatherer groups together with these Neolithic peoples and together with these steppe peoples. And you can also think as the European genetic variation as a melting pot of these sort of three ancestral populations, the original hunter-gatherers, Neolithic farmers, and these steppe herders that moved into Europe later on. And so this is why we can sort of paint the genomes of modern Europeans as this sort of three-way mixture. So every individual in this plot, whether they come from Norway or Croatia or Greece, has some ancestry from this Neolithic group that came from the Middle East, as well as ancestry from this hunter-gatherer group that was in Europe before this Neolithic expansion at low population densities. And the green here represents these steppe populations that moved into Europe later on during the Bronze Age. You can also see that different people from different parts of Europe have more or less of these different ancestry. So, so apparently Sardinians and Albanians have quite a lot of this Neolithic ancestry, whereas Norwegians, Lithuanians, and Estonians have a lot of these steppe ancestry. But all these populations have some amount of these three different ancestries. Okay, so one of the things that tends to happen in my field in ancient population genomics is we can obtain some ancient genomes, and we can compare those ancient genomes to modern genomes, and we will try to paint these genomes in different ways. We've seen many examples of this so far. And then what happens after we've painted genomes on different ancestries, we look at a map and we start to draw arrows on this map. And the reason why we draw arrows is we're trying to explain these patterns that we see as sort of migration processes or population expansion processes that are happening over time. So in order to explain this pattern that we're seeing here, well, we can think of these different arrows moving into Europe, or these different arrows moving into Europe from the Caucasus or from the Middle East. Now, this is quite a strong simplification of this process, but it's quite widespread in the population genetic literature. It's very often that you see an ancient DNA paper or a paper trying to explain the history of a certain group of people, whether that be in Europe or in other places across the world, as sort of being driven by these movements of people depicted by arrows. So you can depict, again, European ancestry as a mixture of these hunter-gatherer groups that moved into Europe very early on, together with these Neolithic groups and these steppe populations. You can also depict sort of the broader out of Africa migration as a bunch of arrows, the colonization of the Americas by Native American peoples, also as a bunch of arrows, or the colonization of different parts of Southeast Asia. Now, this is quite a common practice. I'm guilty of doing this myself. I was involved in some of these studies, like this one, for example. But it has its problems, this practice of drawing arrows on a map. And the first problem is that arrows can be quite ambiguous. Sometimes it can mean that a population has been displaced, so it has moved from one place to another, but it could also mean that a population is just expanding, it's sort of increasing its range. And an arrow is not very good at depicting whether what we're seeing is a displacement or an expansion. Arrows are very bad at reflecting how fast a process is taking place, so how the speed of population movement over time. They're also very poor at reflecting the spatial extent of this process, and they run the risk of overinterpretation, particularly by people that are not working with the data directly. They might just see a cartoon that you've created with arrows, and they might read more into the drawing that you might have wanted to depict. And so I was struggling for several years with this problem of like, how do we better depict population movements rather than just drawing a bunch of arrows in a map to explain the genetic patterns that we see. And so I was looking at this map of Europe, and this is a map that depicts different time slices of Europe over time, going back to about 12,000 years into the past, 10,000 years ago, 7,000 years ago, 6,000 years ago, and so on and so forth, until you get close to the present. And each of the dots in this map represents an ancient genome. This was all the available ancient genomes that were available a few years ago. This has now been populated by many more points since then. And the colors in this map represent how much ancestry from a particular source is present in each of these genomes. So in this case, I'm showing Yamnaya steppe ancestry. This is an ancestry that is particularly prominent in Europe during the Bronze Age. So there's very little of it in the first few time slices, but then at some point in the Bronze Age, you really start seeing it into Europe, and now you see it quite prominently, particularly in parts of Northern Europe. And I was looking at this map, and this map reminded me quite a lot of a different set of maps that I had seen before. This is a different map. This is a map that has nothing to do with genomic data. This is a map of stations that measure particular matter in different towns or field sites in Germany. So it's a measure of air pollution in Germany. And this is a map that also has different time slices. These are measurements that have been taken at different days within the year 2005. And you can see there's quite a lot of changes or fluctuations over time. And it's really a proxy for fluctuations in air quality over time. Now, often what does with this type of maps, and this is a very common practice in the environmental sciences, is do a spatiotemporal interpolation. So this map can be a bit hard to read, and it doesn't tell us much about what's happening sort of in the gaps that we haven't measured. So what we can do is try to interpolate all this data to create sort of a continuous spatiotemporal map of air pollution over time and across space. And so as I was looking at this map of ancestry over time, I was thinking, well, why don't we treat this as an environmental process and use some of the tools that are coming, the statistical tools that are coming from the environmental sciences to better depict these population movements across time and space. And so one of the most common practices in the environmental sciences is something called spatial kriging. And this is just a very fancy word for taking a bunch of measurements in a landscape. And each of these measurements have different colors depicting perhaps a low amount of some quantity or a high amount of some quantity. It could be sort of low pollution or high pollution. And what spatial kriging aims to do is try to predict observations that have not been measured. So what does this square in the map would look like, given the fact that it's close to these two observations and far away from these two observations. And so we can use observations that are close to this point, as well as observations that are distant from this point to make a prediction for what this point would look like, what will be the value of that point. And we can do this for a particular grid cell, but we can do this for all the grid cells in this map to make a continuous interpolation across space. Now, the problem with ancient DNA is that it's scarce in certain periods, while it's more abundant in other periods. You can see that in some of these time slices, there's very little ancient DNA data. Here, there's almost no ancient DNA data in this period, or this period, there's very little, whereas in other periods, there's quite a lot. And this has to do with historical accidents in sampling. So there were perhaps some papers that were really wanted to focus on the Bronze Age, so they generated a lot of Bronze Age ancient genomes, or some papers that really wanted to focus on the Neolithic, so they really generated a lot of data for the Neolithic. While for other periods, there's much less data. And so how do we account for the fact that for certain periods, we might not have any spatial measurements to do our interpolation? Well, the answer is that we can use information from across time slices to try to fill in these temporal gaps. So in addition to doing spatial kriging, we can also use information from cells that are either before or after the observations that we're looking at to try to inform us about what the value of a particular grid cell will be in terms of ancestry. And this is called spatiotemporal kriging, which is just, again, another fancy word for saying we're going to try to predict what an unobserved grid cell is, the value of this unobserved grid cell, by using not just information from neighboring observations, but also information from observations that exist at different time slices. And there's some very, very elegant mathematical theory behind this method, which I'm not going to bore you with, but it has this method has some very nice properties, including the fact that it's the best linear unbiased predictor for an observed grid point. So we published a paper a few years ago where we basically borrowed this method from the environmental sciences and applied it to our ancestry painting predictions. So we took some ancestry inference predictions for different ancient genomes. We put them on a map, and then we did some spatial-temporal kriging on that map to create sort of continuous maps of ancestry movement across space and time. I'm not going to show you results from this paper because since the publication of this paper, we've actually the number of ancient human genomes has actually increased quite a lot. Recently, I was involved in a very, very large study, including thousands of ancient human genomes from throughout Western Eurasia, sampled over the last 10,000 years, which really has helped us create much more accurate predictions of how these population movements have occurred. So what I'm going to show you is results using this much bigger data set, but using the method that I just described. And so what I'm going to show you now is an animation of our best prediction for how the Neolithic expansion, this movement of people from the Middle East into Europe, occurred over time using all these thousands of ancient genomes that we now have available. So you can see that about 10,000 years ago, most of this Neolithic ancestry is concentrated in the Middle East, but over time it expands, and it expands into Europe at different rates and in different ways. But you can see that this much more dynamic way of visualizing how people are spreading across the landscape helps us sort of bypass some of the problems that drawing a bunch of arrows have. Now we have a much better visualization of the speed of this moment and the spatial extent of this moment across time. We can do this with the Neolithic expansion, but we can also do this. We can do the converse and look at this other ancestry of Western hunter-gatherers that were in Europe before these Neolithic peoples migrated into Europe, and you can see that this ancestry is quite prominent at the beginning of the Holocene, but then as Neolithic peoples are moving into Europe, it kind of gets diluted. And so in the present day, there's much less of this ancestry present in present day Europeans. But you can see also that even though it gets diluted much faster in the south than in the north, there's sort of a refugium of hunter-gatherer ancestry in Scandinavia in the late Neolithic. Okay, and another ancestry that we can look at is another group of hunter-gatherers are called the Eastern hunter-gatherers, and these hunter-gatherers contributed a lot to this Bronze Age steppe movement of people that happened after the Neolithic expansion. And so you can see these Eastern hunter-gatherers being quite concentrated here in the Eurasian steppe. During the Bronze Age, you're going to see some spillover ancestry going into Europe, so contributing to European present-day ancestry. And so we can do this with different types of ancestry. We can look at these maps across space and across time, and we did this in this paper. We also have another ancestry I haven't really talked too much about, just this Caucasus hunter-gatherer Iranian farmer ancestry, which also contributed to the Bronze Age steppe expansion. This provides us a better idea of how these population processes unfolded over time and space than just a bunch of arrows. Now, the neat thing is that in addition to genetic data, human genetic data, we also have very detailed paleovegetation reconstructions for these very same period of time. So paleovegetation experts have used pollen data from different sites in Europe over the last 10,000 years to try to reconstruct what the vegetational landscape of Europe looked like at different points in time. Was there perhaps more of a certain type of forest in the early Holocene versus in the late Holocene? Was there more grazing vegetation? Was there more of a pasture type vegetation at different points in time and at different points in space? And we can try to compare these paleovegetation reconstruction maps with the population movements that we observe for humans and see whether there's some relationship between them. So particularly, we can try to see whether changes in vegetation for different types of vegetation can be explained by changes in human population movements, controlling for the fact that the climate has also changed and may have also affected the types of vegetation that you might see in Europe across different periods. And so we develop a model for relating this different types of ancestry, hunter-gatherers, Neolithic farmers, and steppe herders, with the presence or absence of particular types of vegetation in Europe across the Holocene. And one of the interesting things we saw was that the Yamnaya ancestry, the steppe ancestry that came in during the Bronze Age, was particularly positively associated with pasture type vegetation, and negatively associated with certain forest types, suggesting that perhaps as these steppe peoples were moving into Europe, they were perhaps changing the landscape and creating new vegetational environments over time. And so human movements or human expansions had different types of consequences on the vegetational makeup of the European landscape. Another neat thing that we can do is we can compare this expansion process of genetic ancestry over time with reconstructions of the expansion of farming technologies over time. So people in archaeology have been working with trying to see how farming technologies, particular farming practices, established themselves in different parts of Europe at different points in time. And we can see that this is a very parallel process to what we see with genetic data. So farming practices sort of follow genetic population movements, meaning that people that are migrating from the Middle East are bringing with them these different types of technologies to change the landscape. And this sort of feeds into a general understanding, not just of genomic data, but just human life in general, and sort of shows us that genetics has a lot to say about understanding sociocultural processes that unfolded in different parts of the world over time, as long as we can establish close dialogues with other fields that also have a lot of information to provide us, including archaeology and linguistics. And a common language of understanding here is spatiotemporal modeling, or statistical methods that can sort of help us bridge fields that were previously sort of were much more siloed. Okay, so that's all I wanted to say about ancestry. And for the next part of my talk, I want to talk about natural selection in space and time. And so the driving question here is, how can we model the evolution of individual genetic variants? So rather than ancestry, we're now looking at individual mutations in the genome that might have expanded or contracted over time and space. And so just as an overview, what I mean here, natural selection is a process that unfolds over time. And the simplest model that we can think of for how natural selection operates is that we might have a population of individuals carrying different variants. And perhaps at a site in the genome, a variant arises, say a mutation arises in a site that was originally a thymine and then mutates into an adenine. And this mutation might be beneficial. It might confer certain benefits to individuals carrying it, either because of increased viability or increased reproductive rate. And for one reason or another, that mutation over time will expand across the entire population. So what was originally a population that had a majority of this T variant, after several years or several generations, you might find that same population now has a majority of the A variant rather than the T variant, because the A variant was perhaps beneficial or selected for. Now, this is a process that occurs across time, but it's also a process that unfolds across space. And so here's a depiction of a mutation that appears at a particular point in space, somewhere in Eastern Europe. And because this mutation is beneficial, eventually it might expand over time and sort of cover a much larger area than it covered before. And this is the process that I'm interested in modeling, understanding how we can account for the fact that we're not just looking at a temporal process, we're looking at a spatiotemporal process that unfolds. Several people have worked on this problem before. There was a paper in 2005 trying to model the spatiotemporal evolution of a variant in the CCR5 gene, which has been associated with HIV resistance and was probably associated with resistance to pathogens in the past. And for this, they use what is called a diffusion equation. And I'm not going to bother you with the details of this equation, but I just want to mention the fact that within this equation, there's a particular term called sigma that sort of controls how fast this diffusion process unfolds. And there's another term that controls local growth at a particular region in the world. These two terms are important if we want to account for not just the increase in frequency of an allele, but the fact that the increase in frequency is happening across a landscape. And so what I'm going to show you now is work by one of my PhD students, Rasa, who extended the model that was originally developed in this paper to not just account for the fact that a mutation may diffuse across a particular latitudinal and longitudinal dimension, but she also introduced what are called advection terms, and these allow the center of mass of this variant to differ from the allelic origin. And this is just a way to accommodate the fact that a variant may not just expand across the landscape, but it might also be displaced over time because of population movements. And she also did some neat work to adapt this methodology for ancient genomic data, which has a lot of idiosyncrasies. It's quite hard to work with ancient genomic data because it tends to be quite fragmented. It tends to has a lot of gaps, and so she did some statistical work to adapt some of these methodologies to work quite well with ancient genomic data. And so she developed a model that can trace the evolution of a variant across time and space. And so to show that the model works quite well, here's a simulation of a variant expanding across the landscape over time. And this is what Russ's method infers across different time slices. You can see these two trajectories are quite similar to each other. And obviously, how accurate her method is depends on how much data we have. And so we're going to focus now on ancient Europe because there's quite a lot of data in ancient Europe to make very, very accurate inferences of this spatiotemporal process. So she used all these ancient genomic data, from which we have quite a lot of genomes, and she also used some present-day genomic data from across Europe and parts of Central Asia as well. And so she was tracing the evolution of particular variants that are known to have been under selection. This is one example. This is a TYR, an allele in a gene called TYR that controls skin pigmentation, eye color, and that is fairly infrequent in the early Holocene, but then starts to appear in parts of Europe and eventually expands across Europe. And this is Russ's method's reconstruction for how this process unfolds. You can pinpoint the origin of the allele to be somewhere here, and then the expansion happens over time. And you can see that the places where the allele is now most frequent is not necessarily the same places where the allele may have originated. She can also provide estimates of how strongly selected the allele was, and look at differences in the strength of selection at different periods of time. She's also done this for a very famously selected allele, the lactose persistence allele. This is the allele that allows adults to digest lactose, and that is known to be quite high frequencies, particularly in Northern Europe, whereas in Southern Europe and other parts of the world it's at lower frequencies. And so she can reconstruct sort of the spatiotemporal history of this allele, which has an origin somewhere here, perhaps somewhere where we had steppe populations in the early Bronze Age, and then expands over time and also moves to the west over time. And she also predicts that there's quite strong selection operating on this allele. So her method is publicly available. You can check out her paper on the bioRxiv preprint server if you want to learn more about it. The lactase persistence allele is quite a well-studied allele. It has a very interesting history. I'd like to focus a bit more on this allele because of work that another person in my group, Evan Irving Peace, has done. We can look at the rise in frequency of this allele in Europe over time. This is a very neat reconstruction of how this process unfolded over time in Europe, and you can see that the largest period of the strongest period of rise in Europe is fairly recent, at least by evidence by this figure. What you're seeing here is in the x-axis time, so thousands of years ago into the past. The y-axis is the frequency of these allele. You can see it's increasing, and but you can also see that this is a bit cloudy, and the reason for this is that there's some uncertainty around the exact allele frequency at a particular period of time. So this cloudiness reflects that we're not extremely sure of the frequency, but we have a fairly good estimate of where the frequency was. And so one of the neat things that Evan has been working on is trying to separate the rise in frequency of selected alleles in different ancestral populations. And this is work that he's done in collaboration with Richard Durbin and Alice Pearson, as well as Rasmus Nielsen and Aaron Stern. They're in Cambridge and in Berkeley. And what they've been trying to do is trying to see whether we can separate these different selection processes in the different ancestral populations that we know make up modern Europeans today. So remember that I told you before that in order to sort of model present-day European genetic variation, we had to account for the fact that there was this Neolithic component, this Western hunter-gatherer component, this Eastern hunter-gatherer component, and another Caucasus or Iranian Neolithic component as well. And the question is, was this rise in frequency that we see when we just look at all the ancient DNA that we had, was this process unfolding in a similar fashion in all these different populations, or was there perhaps stronger rises in frequency in one of these populations versus another? And Evan has done quite a lot of work to try to separate these processes. And one of the things that you noted is that when we only focus on these Neolithic Middle Eastern farmer populations that came into Europe during the Neolithic, we don't see a very strong rise in frequency until fairly recently. And when we look at Western hunter-gatherer populations, we also don't see a very strong rise in frequency. Whereas when we look at Eastern hunter-gatherer populations, we actually see quite a strong rise in frequency, and it's a rise in frequency actually starts earlier than when we just look at the entirety of the data. We also see a similar pattern when we look at this Iranian hunter-gatherer ancestry. And what this is telling us is that the LCT allele, this lactase persistence allele, may have been under selection earlier than previously thought, particularly in populations with a steppe or central Eurasian ancestry, which expanded into Europe during the Bronze Age, suggesting that in different populations during the Holocene, this allele was under more or less different selection pressures over time. Evan's analysis is part of a very big study that by Morten Alentov and Martin Sikora and Eske Willerslev, which is now published as a preprint on bioRxiv, and you can check it out. It's one of many other analyses that have been done on a very large scale data of ancient genomes from throughout the Holocene, particularly focusing on the Mesolithic to Neolithic transition. So I encourage you to take a look at this manuscript. I also encourage you to take a look at another recent paper on the rise in frequency of the lactase persistence allele. This is a paper that was recently published a few months ago, which tries to look at what was the main driving force behind the rise in frequency. So what was the likely cause for this rise in frequency? And it has some surprising answers, and it suggests that it may not have been the presence or absence of dairy farming in different parts of Europe that was the main driver of this frequency, but actually maybe changes in demography and changes in population density that was a stronger driver for this rise in frequency. It's a very interesting paper. Okay, how am I doing with time? I'm doing pretty okay. Okay, for the third, and maybe I'll take a moment here to just check on the class and see how you guys are doing. Do you guys have any questions about the first two topics that I talked about? I know it's a lot of information. So how are you guys doing in the classroom? Maybe it's a good time to ask if you guys have any questions about the different topics that I've talked about so far.
Guys, if you have questions, please make on the microphone. Where is
yes,
the
there's
microphone?
quite a lot of questions. Yes.
And people who are online, if you want to make question, I can read them for you. Yeah, who is going to?
So let's take a couple of questions.
Fernando, can you explain? I mean, for me, it's hard to understand like how, for example, the lactase tolerance mutation, it appeared like not so long ago, and now it's so prevalent in Europe. And I think it's a useful mutation, but I can't understand like how it can be so prevalent nowadays.
Yeah, so lactase allele it's an exception more than a rule. It's sort of its historical rise in frequency. It's actually one of the strongest selected alleles in human history, particularly in recent human history. The reason why it's so prevalent is because it was under very, very high selective pressures, meaning individuals who carried this mutation had a strong advantage over those that did not carry it. Now, the reason why this advantage that is quite under contention, and it's part of the topic of this paper. And so one of the traditional explanations for this is that if dairy farming became widespread fairly recently, then if you could digest milk, then perhaps into adulthood, perhaps you were more likely to acquire nutrients or more likely to survive than if you weren't able to digest lactose into adulthood. And what this paper argues is that might not be true. There might be other processes happening, and that this variant may have been in their selection for other reasons, including response to pathogens or changes in population density over time. To really understand the cause of this, I think you would need a time machine, but I think this paper tries to narrow down on the actual cause. It's still very hard because it's something that has happened in the past. We can't go back, but you can try to see, for example, whether in those parts of Europe where there was a strongest rise in frequency, if those parts of Europe were also the ones where milk production or dairy farming was strongest. And what this paper argues is that that's not really the case. There's some places in Europe where you saw strong rises in frequency, but there's very little evidence for dairy farming. And so the explanation might lie somewhere else.
Thank you very much.
I think we have another question.
Well, my question is, how can we track the climate conditions from a long time ago?
Mm-hmm. Yeah, so for that, I think, so I'm not a climate scientist, but the way it essentially works is by climate modeling and a lot of simulations. So one can study patterns of climate change in the recent past and project into the future under certain conditions. Another big source of information is also pollen data. So pollen data can reveal whether some regions of the world were under higher or stronger fluctuations in temperature and precipitation, which also affect the vegetational landscape. Yeah, I don't have the strongest expertise on climate modeling, but I know a lot of it is based on projecting fluctuations in climates that we see now into the past using simulations.
Thank you.
Okay.
Just a quick reminder to the online people, if you want to make questions, just type them and I can read it for Fernando. Okay? Yeah, you can continue, Fernando.
Super.
Thank you.
All right. So the third topic I want to talk about is simulation. So simulating genetic data in space and time, which is an increasingly strong focus in my research group. And the question is, why simulations? Why do we care about generating simulations of population genetic data? And there's many reasons why simulations are useful in population genetics and evolution genetics in general. So the simulations, for once, can provide us with strong intuitions about how genetic data will behave under different types of demographic models. So, for example, it can allow us to understand what happens to a population when it undergoes a bottleneck, or when it undergoes an expansion, or when it moves from one place to another, and can then allow us to see, well, if we see these patterns that we see in the simulations in real life, then we can sort of reconstruct what may have happened in the past. So it provides a good way to build intuition about the behavior of genetic data. Another reason why simulations are important is because they allow us to test inference methods. So if we have a set of methods for inferring how natural selection unfolds over time, or inferring patterns of demographic history, and we want to tell, well, how accurate are they, which of them I should use for a particular problem that I might have, it's good to have simulations, because simulations sort of provide a ground truth on which we can evaluate the accuracy of these different methods and say, well, if I know that my simulation unfolds like this, and then I try to infer it using method A, I get quite a crappy prediction that's very different from what I simulated. Whereas if I use method B, I get quite a good and robust prediction. So when I try to study real data, I'm rather going to go with method B than with method A. Another reason why simulations are useful is that they can also be used for inferring demographic parameters, so inferring population histories in the past. There's a method with a fancy name called approximate Bayesian computation, in which one generates a shit ton of simulations, so thousands of simulations under different demographic histories. Some say, for example, some might include a bottleneck, some might not, some might include a population expansion, some might not. And then one computes summary statistics from these different simulations. So computes summaries of the data that you see in the different simulations, and then compares those summaries to what you see in real life. And so you try to see which are the simulations that best match to what you see in real life, and that allows you to obtain parameter estimates. So there's quite a lot of uses for simulations in population genetics. And the question that we were interested in tackling in my group is, how can we easily simulate spatiotemporal genomic data? And the reason for this is there's quite a lot of methods for generating genomic data in this kind of like tube-like models, where space is completely ignored. So this is a representation of European history that completely ignores space. It just accounts for time. It says that at some point we had an African population. Later on, this African population expanded out of Africa, and so it's labeled as an out of Africa or OOA population. At some point, this population diverged into multiple populations. So, Anatolian farmers, Eastern hunter-gatherers, Western hunter-gatherers, Neolithic farmers, Yamnaya steppe herders, and eventually these all combined together to make up what we have in terms of genetic variation in Europe today. And there's quite a lot of methods for easily generating simulations when you just treat populations as if they exist in a sort of void, a non-spatial void, just accounting for the fact that sometimes they split and sometimes they mix together. But geography is intrinsic to our past. We've seen in a lot of our examples, and so ignoring geography can sometimes lead us astray. So it's important to account for the fact that as these divergence and mixture processes are happening, they are happening on a landscape. They're happening on some spatial dimension, and they might happen in different parts of space at different rates or in different ways. So really accounting for space in simulations is really important. What I'm going to show you is work by one of my postdocs, Martin Petter, who developed an R package to very easily build spatiotemporal genomic simulations. So to create populations that actually live in a geographic landscape and that might move or expand or contract over time. And as part of this package, he's created a lot of different R functions that can allow biologists or users of this package to very easily define a population and its spatial extent and how it changes over time and space. And I'm going to show you some examples of what can be achieved with Slender in the next few slides. So let's say I'm interested in modeling a population or simulating a population that exists somewhere in Eastern Europe, as depicted here. And we're gonna call it YAM. And this population has a certain spatial range that's determined by these vertices here. And in Slender, in this R package created by Martin, it's very easy to define this population and say, well, it exists under a given certain coordinates that define this polygon. Its target existed at a certain point in time, and it had a certain population size. In this case, it had a population size of 600 individuals. It also can very easily specify that this population may have originally come from some other population that was previously simulated, and this is what stands here for the parent population. And you can see that with a very few lines of R code, you can create a simulation or specify a simulation of a spatially existing population. The nice thing about Slender is that it can also allow us to say, well, this population, somewhere between 6,000 and 3,000 years ago, this population moved westward into Europe. And again, with very few lines of R code, we can say the trajectory that it moved is like this. It's this started at this point in time, it ended at this point in time. And so with very few lines of code, we can actually specify this population movement. And the nice thing about Slender is that it sort of hides away or takes care of all the nasty details that come with space. So you can see, for example, here, even though I moved this polygon from here to here, Slender accounts for the fact that a population is not going to exist in the water. So it doesn't simulate anything that is happening in the water. It just assumes that a population is moving across space. And so it takes a lot of care of all the nitty-gritty details of space that you don't want to necessarily care about when we're simulating genetic data. Slender can also very easily simulate a population expansion. Here, for example, simulating the expansion of Neolithic peoples into Europe over time. And this can just be done with a very simple R function called expand that says, well, I'm going to expand this population by 2,500 kilometers squared, and it starts at this point, it ends at this point, and I'm going to do it over this particular region here. And again, you can see that the simulation program is taking care of the fact that you might encounter land barriers, and you shouldn't extend your population into the ocean. So here's a full simulation of spatial-temporal dynamics in West Eurasia. It's a toy simulation. It's a very much a simplification of what really happened in Europe over the last 10,000 years. But it sort of illustrates how, again, with very few lines of code, we can simulate the fact that you had some original African population. This expands somewhere into Central Asia. And then you create some additional populations like these Eastern hunter-gatherers, Western hunter-gatherers, and farmers. Then you have an expansion from Anatolia and a population movement during the Bronze Age. And so each of the dots that I'm depicting here represents an individual that exists at a particular point in time. And importantly, the simulation can give you the entire genome of each of these individuals. So if I want to study the genome of any of these individuals in the simulation, I can directly obtain it from Slender. Slender works as a wrapper of another simulation program called SLIM, which is doing a lot of the backend work for simulating genetic data here. And it can handle a whole suite of spatial-temporal models, not just models like the one I showed here, but also what are called isolation by distance models, where you have a bunch of demes and they exchange migrants across space, as well as different models containing different amounts of clustering or competition and dispersal over time. So there's really a whole suite of things that you can do in the simulation program, and I really encourage you to try it out. You can go to this website called slender.net. Slender.net, and it contains a lot of tutorials that Martin very painstakingly wrote to teach you how to very easily generate a population genomic simulation, even if you don't have any previous experience with genetic simulations in the past. So I really encourage you to try it out. So it's now on CRAN. So you can easily download it from CRAN if you have an R installation, and it has a ton of different vignettes with tutorials about how to generate population genomic simulations. In terms of future directions, we're going to aim to incorporate geographic features into simulations as well. We're going to do continued work on mobility, migration, and cultural change and their interactions. We're going to try to see whether we can correlate ecological and environmental factors affecting mobility in the past, and see whether we can also incorporate other types of non-genomic data like isotope data, vegetation, and linguistic data into a spatial-temporal inference framework. With that, I'd like to thank a lot of people that collaborated in a lot of these projects. Particularly Martin, Russ, and Evan, who drove a lot of the work that I showed you today, as well as all the members of my research group, which are depicted here, including the newest member, Matthias, my baby son, as well as funding institutions that provided funding for this work. And we have half an hour, and so if you paid attention, you know that I have one more topic that I want to talk about before I finish my talk. That's what I'm going to do in a second. Before I do that, I just want to check with the classroom if there are any questions about the last section on simulations. So I'm just going to just do a check if anybody has any questions about simulating spatial-temporal genomic data.
Do you want to make questions, guys? We have a question here. Just a second.
Yeah.
Hi, I was a little confused about what precisely in what you showed constitutes a simulation, because I felt like it was the exhibition of data in a way which will consider the space which already exists in the data you have, but and from a simulation of this, I would understand something like setting a certain distribution of the alleles in space and giving conditions for its evolution in space.
So that's a good point. So what I'm showing you here is sort of like a geographic depiction of the simulation, but I'm not showing you what is actually coming out of the program. And what is coming out of the program, so this is what you specify in terms of geography. What is coming out is that for each of these dots that you see here that existed at some point in space and time, what you get is a full genome from each of these dots, so a full sequence of A, C, T's, and G's. And that you can treat as a human genome or as a genome of a particular organism that exists in space. And then you can do whatever analysis you would do with genomes in real life. So you can compute summary statistics, you can try to infer patterns of ancestry from them. So each of these dots is really a large sequence of nucleotides that sort of represents the fact that this is an individual that is evolving on the landscape. So if you're interested in seeing, okay, what would genetic variation look like if a population moved from east to west or a population expanded? Well, all you have to do is just take one of these dots and you have the simulations from each of these dots and just compare them with each other. So what you obtain from these simulations is a full genome, so a full sequence of nucleotides that has evolved under the particular conditions that you simulate. Does that answer your question?
Somewhat. So it's an agent-based model, a simulation which is being done, and are the agents reproducing between themselves, or are they answering simply to a step function?
No, yes. So each of these individuals is an individual that is reproducing under something called the Wright-Fisher model. So each of these individuals at each generation is a descendant of individuals at the parent generation, and so on and so forth. The nice thing about the framework is that the generations can exist in space. So if you have an individual here, it's likely that it came from some individual close in space. And you can define the populations like, for example, you disallow individuals that are here to have descended from any individuals, say here, if there's some sort of reproductive barrier between the populations.
Thank you.
Thank you.
Do we have any
so
other questions?
There's the full genealogy linking these different individuals together.
Okay, but the evolution of populations is always an input that you give, like migrations and the like
you specify things like migration and population expansion. You can also do, I mean, if you don't want to be if you don't want to specify a lot of different parameters, you can also just have a population evolving under certain conditions. Like, for example, say I just want to simulate a population in Europe, And I want to say individuals can freely migrate, but only up to a certain distance, or only produce children within a certain radius. And so you can specify more ecological parameters like that, or you can be more sort of micromanaging of your simulation and say this population exists here and then it moves to here.
Okay, thank you.
Do we have any other question before he continues? I don't think so. Fernando, you can continue to the topic, magic topic.
Yeah, the last topic that I want to talk about is something that I've been increasingly involved in maybe the last 3 years. It doesn't have a lot to do with genomics, but it does have a lot to do with research and science in general. And I want to talk about a preprint of a paper that I recently published together with a group of biologists and ecologists. I published it just a few days ago, last week. And it has to do with the role of life scientists in the times that we live in. So a lot of you are biologists or ecologists in training, and I think this is particularly important for you guys. And it's a paper about our role in the current climate and ecological crisis that we're all living through. And so how we engage with the public in this sort of planetary crisis that we see ourselves in. Now, a lot of you probably know the fact that carbon dioxide concentrations are skyrocketing, and they're causing our planet to increase in temperature, and quite dramatically over the last few decades. So right now, concentrations of carbon dioxide are around 420 parts per million, and since pre-industrial times, our Earth has warmed by about 1.2 degrees Celsius. This is sometimes in the news, perhaps not as often as it should be, but you should probably be familiar with this. And this is where we are now in terms of warming. This is from an article published in PNAS in 2020, and this shows different areas of the world and how suitable they are for human life at this point in time, with the amount of warming that we've had since pre-industrial times. And what you see here is, high regions of high suitability for human life are in light colors, and for low suitability, you can see here, are in darker colors like Siberia, Greenland, or North America. The black areas here, the big regions that are basically unsuitable for human life. It's just too hot for people to live there for prolonged periods of time. This is where we are now in 2020. Under our current projections, or where we're heading in the next few decades, by 2070, this is what this map will look like. So about 20% of your surface will be unsuitable for long-term human survival, including large parts of your own country, of Brazil. And this will affect about 3 billion people. So it's a crisis of extraordinary proportions that we're living through, including many areas that are producing food for many people, even those areas that will be still suitable for human life. So we're really living through a big crisis at the moment. And this crisis is already manifesting in terms of droughts and massive famines across the world, fires, as well as massive floods across the world, and consequent displacement of many people across borders, and generates conflict when people are forced to be displaced and leave their home. Really, scientists have been warning about this for many decades now, about this process unfolding in our entire planet. But really, we have failed to activate society in the right way. So there's been many reports about climate change. There's been many agreements and accords and conferences trying to deal with this problem, and yet the problem has only been getting worse and worse over time. And so we find ourselves in this sort of exponential curve of planetary destruction, and projections for what's coming in the next few decades are grim, to say the least. And some scientists are now arguing that we should be better at communicating the crisis, and we actually should be acting on our own warnings to humanity. This is a quote from one of these papers that was published in the journal Nature, Ecology, and Evolution a few years ago, saying that we can no longer carry on as normal. Acceptable forms of influence are not working, and we are running out of time. The scientists who alerted the world to the climate and ecological crisis have a moral duty to join the popular movements demanding political action. This is another article arguing that scientists should take more active roles in warning or engaging with the public by facilitating academic advocacy and activism in the climate and ecological emergency. And this is another article published in the journal Nature just a few days ago, arguing that scientists engaging in civil disobedience can help press for urgent climate action. And so increasingly over the last few years, I've been doing that. I've been participating in activism and scientist activism by engaging in acts of civil resistance, for example, blocking streets or occupying buildings to warn governments, in my case, the Danish government, of how little action is being taken to prevent the worst consequences of the climate and ecological crisis. Obviously, we're not alone here. There's many scientists across the world that are doing this, including in the UK, in Italy, in the US. Scientists have been chaining themselves to the White House. In Nigeria, in Spain, many scientists were arrested for warning about this and occupying the main government building in Madrid. In the Netherlands, they occupied a fossil fuel company. In Germany, they were blocking roads. In my native country of Argentina, there were different teach-ins on the climate and ecological crisis, as well as in Norway. All of this happened in a single week, just this April, where scientists across the world were saying, publishing papers is not enough. There's more that we need to do. We need to better engage with the public by engaging in activism. I encourage you to check out this paper because it touches on a lot of these topics, and it particularly on our role as life scientists, as biologists and ecologists in the biospheric emergency of the climate and ecological crisis and what we can do about it. One of the figures that we show in this paper is that activism by scientists is not new. It has a long and rich history extended for many years, including in Brazil. There were a lot of scientist activists that were instrumental in environmental protection policies in the first half of the 20th century, biologists working at the National Museum in Brazil. And scientists throughout history have pushed for many policies, including the women's vote movement, social justice, and the anti-war movement, and increasingly now for climate and ecological justice. And the main point of the paper is that our traditional roles as scientists should be expanded. So we usually think of the role of scientists as involving engaging in research, engaging in teaching, and engaging in outreach. There's a lot of things we can do to expand the normal practices or the normalized practices that we currently think scientists as having by engaging in acts of activism and civil resistance in whatever form we think appropriate. So the take-home message from this last part of my talk is that cost some trouble, engage in activism, and try to do it together. I'm sure there's many climate direct action movements in Brazil that are currently active and need as many people as possible. And if you're a researcher, an academic, or a university student, I encourage you to join a Scientist Rebellion, which is a movement of scientists engaging in civil resistance. And with that, you can check out the website for Scientist Rebellion here. With that, I'd like to thank you for listening, and that's the end of my talk. Are there any questions?
Thank you very much, Fernando. I guess maybe we have some questions coming from the people online or from the classroom. Does anyone want to make a question? Brave guys. So I have a quick question about this last topic. I'm curious about how is that? So you're doing this in the Denmark, right? Did you do this in Argentina already, or it was how?
Yeah, so I mean, I haven't lived in Argentina since I was a teenager. So,
Okay.
but I know there's scientists in Argentina that are engaging in activism as well. So that was one of my examples. And obviously, different countries have different amounts of scope for what you can do. There's some countries that are much more repressive than others. So the type of action that you can do in different countries varies. And so that's something that we always keep in mind, that, you know, it's much less likely that something very, very violent might happen to you with respect to police action in particular countries than in others. And I would say the stark example here would be like, you know, in Russia, for just holding a sign that says, you know, peace, you might get arrested for several days. So I think it's important to account for the fact that in different countries, the legal situation and the repressive situation is very different. But what we try to encourage is, like, from whichever point in the world we are, to try to do some type of action to better engage with the public, to try to generate some media attention about the crisis and try to break through the sort of politically established silence that exists about the crisis.
Oh, okay, great. I was curious if you had any kind of violence while protesting.
Yeah, so I mean, a lot of the actions that happen in countries in the south of the world, particularly in places like Africa and South America, were fairly non-illegal. So it could just mean, you know, going to a park and just have an open classroom about the crisis, perhaps in front of a government building, without actually blocking a street. I think this is what happened in Argentina, for example. So it depends a lot on the frameworks of each country and the amount of police repression in each country. But each of the different groups that are already operating, scientist actors that are operating in different countries, have different tactics for attracting media attention. So what I would encourage you to do is contact other scientist activists within the movement and see what they are doing to try to get some attention. Cool.
And how did people in Denmark receive you? Like the people.
So yeah, so I mean, I've been involved in several actions in Denmark. So I've been in some actions where the police just let us be and just let us block the road for several hours. I've been in other actions where the police detained me for some time. And I've seen some of my scientific colleagues get arrested for a few hours. So it really depends a lot on how happy the police is and the type of action that we're doing. I think it's about sort of, you know, putting a bit of more meat in the game, but obviously within your own limits. So if you don't think you want to put yourself in an arrestable position, all these movements require a ton of non-arrestable support, like roles that don't involve putting yourself in a vulnerable position, but that they could involve helping people put themselves in vulnerable positions. And this could include, for example, filming these events, or helping with logistics, or helping with dissemination of the event. So there's quite a lot of roles that people can take without actually being on the street themselves.
Yeah, but my question was, do the people that are in the street, like while you are doing an event, do they come to ask questions and to interact with you?
Yeah.
How is that?
Yeah. I mean, there's different types of responses. Sometimes, you know, obviously you have people in cars get angry that you're blocking the street, but you often have people that are walking or nearby that want to engage, and you have dedicated roles for that as well. I would say a lot of the response usually comes afterwards via the media, where we've had actions where we had lots of media interviewing us and like allowing us to go on their news channels to explain why we're doing this. And I think that helps a lot with spreading out the message beyond just the people that might see the event on the street at the particular time that we're doing it.
Oh yeah, cool.
Oh.
Thank you. I think there's two more questions. Go ahead, Kyle.
I was checking the website and I saw, just briefly, I couldn't read really well all things, but I see you mentioned economic degrowth, and how could you talk a bit on how that topic is being handled, and how what are you talking about that?
Yeah, so degrowth is an increasingly popular economic idea, which argues that countries that are already quite rich and that don't need to grow anymore should actually implement policies to stop growth, particularly the growth of industries that are damaging to the environment, while allowing other countries that might need to grow or might need to reach particular levels of supply of basic services to still grow. But the idea behind it is that the reigning economic ideology shouldn't be that every country should grow at high rates. It should be that we should be agnostic to growth. If there are some things that we need, like for example, we need to expand our hospital systems, or we need to expand public transportation, those are things that should grow. Whereas if there are other things that should not grow or should actually degrow, like the fossil fuel industry, the amount of cars on the street, those things should be scaled massively down. So it's a movement, it's a sort of a movement that is becoming increasingly popular, particularly for addressing the climate ecological crisis, putting a lot of attention on climate justice, on the fact that most of the responsibility for the climate crisis is on rich countries, particularly in the global north, and particularly rich people within those countries. And so accounting for that, figuring out ways in which we can decrease our impact on the environment and try to make space for other people that are not contributing to the crisis and that still want to have a good life to live.
One more question.
So hello, I wanted to ask about the last part of your presentation, and thank you for the lecture. It was really interesting. And we are having elections this year in Brazil, and there is a movement of scientists that are trying to be part of the political positions and to try to be a part of the Congress. And so that's one of other ways of activism that we can do as scientists. And I wanted to ask you if there is something similar happening in Denmark, where people are trying, the scientists are trying to be more a part of the people who make the decisions.
Yeah, so I think it's good that scientists are becoming more involved in politics. I would say for a lot of these problems that we have, climate crisis, which are so much partly a product of representative politics. One of the things that a lot of these climate movements are arguing is that it's not a matter of just having the right politicians in power or electing people that will care about the crisis in coming decades, but it's about empowering people that are not politicians to actually have a say in their future through more direct or deliberative modes of democracy beyond just representative politics, which is been really very much a failure in terms of addressing the crisis. So telling people that there's other ways in which one can contribute to democratic processes beyond just waiting every 4 years to vote for somebody that might not even represent them very well. And so a lot of these movements are pushing for things like what are called citizens' assemblies, which are a form of deliberative democracy to solve very specific problems like the climate crisis, and empower people to contribute with solutions to this. I know in Denmark there's a few politicians that are also scientists, but I don't think in particular in Denmark, there's a strong movement for having scientists in politics. I think there's a very strong movement for scientists and non-scientists to create these alternative democratic bodies to address the crisis beyond just having particular representatives that are elected every few years.
Okay, any other questions? Comment? Another one from Kawe, sorry.
I could follow as a follow-up in the previous question. Well, asking mostly because you are in Denmark, I don't know how spread in Europe and other countries the movement of which you talk is, but generally when we here in the global south, when we think about countries such as Denmark and its neighboring countries, we think that there is big and multinational corporations which enjoy law liberties in other countries and whilst in Denmark, where the state is rather strong, they operate in correct and good manners. So, and one of the things that you say, I mean in plural, dreadful language which does not distinguish plural, say in the site is about degrowth without decreasing the standards of living in those countries. So how is that playing on? Because it seems to me like a rather difficult point to make in such a country like this, and so.
Yeah, I mean, I guess addressing your first point, I think it's very true that a lot of these companies sort of have quite a lot of restrictions within Europe, but then are allowed to wreck the world everywhere else. And a lot of the targets of a lot of these actions are companies like that. So just as an example, this is actually the ones in Italy and the Netherlands. There were two fossil fuel companies that, within these countries, they behave very well, but they are destroying environments in Africa and South America and in Asia. I would say, in terms of how degrowth can achieve this, I would say the point of degrowth is that a lot of the things that we enjoy, like family and leisure and the good things in life, currently are facilitated by fossil fuels. If I want to go enjoy the beach, I have to take a car and drive sometime. Whereas we can degrow our economy while preserving things that contributed to a good life if we invest in the right things. So if I didn't have to take a car, but there was a much more investment in public transport connected me to a beach or a forest. Or if we didn't populate cities with highways and streets, but allow for a lot more living space for people to develop, you would still be able to enjoy the same things you enjoy. You just enjoy them through different means that would involve the burning of fossil fuels. I think this is particularly a problem in the global north, which is so dependent on fossil fuels, but you also see it in countries in the global south. Thinking about Argentina, where there's a very strong car dependency for a lot of things that involve going from one place to another. You need to get a car, even though you might be in a highly densely populated area that could be easily populated by public transportation. So it's about thinking of not just about the things that make up the good life, but also the means that we use to attain those things and how they could be replaced in a much more decarbonized world.
Any other comment? Oh, thank you. Sorry, go ahead. Any other question? Any other comment? Just let me check the online people. So I think that's it. All right. Thank you very much, Fernando, for giving the class yesterday, for the seminar today, and thank you for the people that were online as well. And I hope we can meet you again in next editions of the program.
Sounds good. Thank you so much for the invitation.
Thank you.